In this video on gene expression regulation or transcription regulation, you will learn everything you need to know about the tryptophan operon, which is a regulatory system in bacteria. We will look at how tryptophan operon works in two different bacteria, one being E. coli and the other one is Bacillus subtilis. The trpOperon or tryptophan operon is quite unique in the sense that it is a repressible type of operon. This means that the substrate, tryptophan, in this specific operon inhibits transcription. More specifically, the substrate binds the repressor, and this repressor then inhibits transcription. This is in a strong contrast to the lac operon, where we saw that the substrate allolactose binds the repressor and inhibits the repressor. But instead, the whole process activates the transcription. And lac operon, as we have discussed in the previous video, is a type of an inducible operon. I will contrast the trpOperon with the lac operon just for a while to give you an idea of the comparison. So I hope you have understood lac operon before watching this video. Also, some details about the repressor and operator binding were covered in detail in the lac operon video, so I will not go into those same details again in this video. Alright, in contrast to the lac operon, the trpOperon has a different mechanism of action. We will see that there are some secondary structures in RNA that form, and those influence the movement of ribosomes and RNA polymerases on the RNA. This means that the effect and the regulation is also done at a translational level, which is not seen in the lac operon. The concept of RNA structure and ribosome or RNA polymerase interaction is very similar to riboswitches, where the binding of a ligand changes the RNA secondary structure and that affects the gene expression. trpOperon also has a repressor, this guy that we talked about earlier, which also recognizes operator sequences. But these operator sequences are spread out among 3 loci across the genome in the bacteria. So what are these 3 loci? The first is the repressor locus itself. So essentially, the repressor protein binds its own locus and regulates itself via a negative feedback. And we will discuss how this works very soon. This is in strong contrast to the lac operon's repressor, where the repressor is not regulated at all. The second loci that it regulates is the trpOperon itself, which has 5 genes, E, D, C, B, and A. Remember that lac operon only had 3 genes named Z, Y, and A. The third locus that the trprepressor binds to is the ARO-H gene in the ARO-operon. The ARO-H encodes for an isoenzyme required to make 3 aromatic amino acids, which are tryptophan, phenylalanine, and tyrosine. So there is some shared functionality here. Since we're talking about the synthesis of amino acid, the key point to note is that the tryptophan E, D, C, B, and A genes are anabolic in function, which means they synthesize tryptophan. Just for comparison again, the lac operon is a catabolic operon in function, because it breaks down lactose into simple sugars. Alright, let's actually get into the discussion about tryptophan operon. Oh, just a word of note that the ARO-H is not directly linked to the tryptophan operon, so we won't be discussing that in this video. So let's start our discussion with the trpR locus, which contains the repressor gene called trpR. Upstream of the trpR is the operator region that contains the transcription start site. And then upstream of that is the promoter region. Unlike lac operon repressor, which was sitting next to lac ZYA, the tryptophan repressor locus is located far away from the tryptophan operon. So in the default state, when nothing is bound at the operator, the RNA polymerase will be recruited at the promoter and it makes the RNA. And this RNA gets translated into proteins. And the protein is the monomer, which is similar to the idea of monomer we discussed in the lac operon, with a DNA binding domain, oligomerization domain, and a core domain. Using the oligomerization domain, 2 monomers come together and form a dimer repressor. But this dimer is inactive, which means it cannot bind the operator sequence, and that's the default state. In contrast, if you recall, the lac repressor is a tetramer, and it is active in the default state. The second locus, the interesting one, is the trpOperon itself. In this operon, you have 5 genes named A, B, C, D, and E, and they form the structural genes. And technically speaking, they are metabolic genes. Now here is where things get interesting. Just upstream of the trpE gene is a short sequence of a uracil repeat, which is called an attenuator. More on this later. Upstream of this attenuator is a sequence which contains a leader peptide. Upstream of the leader peptide is the operator sequence, which contains the transcription start site. And then you also have the promoter. In the case of tryptophan Operon, the promoter is actually a strong one, in contrast to the promoter that we saw in the lag Operon. Just for the sake of information, this leader peptide region and until the attenuator together is known as the leader region. It is essentially a fancy name for 5' UTR. The structural genes are the main genes, which are transcribed into a polycistronic mRNA that gets translated into 5 individual proteins. If you're curious about these genes, the trpE and D encode for 2 subunits for an enzyme called anthranilate synthetase. The trpC encodes for phosphoribosylantranilate anthranilate isomerase. Then finally, trpB and A encode for the subunits of the enzyme tryptophan synthetase. I will not go into further details about these genes, but all you need to keep in mind is that together all these 5 genes function to produce the tryptophan amino acid, hence the tryptophan Operon. The interesting bit about this Operon though is this leader region. So let's actually zoom in and take a closer look at it. So the operator DNA sequence has this transcription start site which contains the +1 base that becomes the starting point of the RNA. And then there is a Shine-Dalgarno sequence. At around +26 position from the +1 base is the start codon UGG. So now I'm writing out the RNA. To get an idea of the DNA sequence, you just turn the uracils into thymine. Since most of the magic happens after the TSS in the RNA form, I will write out the sequence of the RNA that forms after this +1 site. So the AUG codon is the start site. And then some codons follow that, and at some point you get these UGG codons. 2 of them back to back. And these UGG codons specify the amino acid tryptophan. And after the UGG codon is the stop codon UGA. And this is somewhere around +68 position. So this region from +26 to +68 is a short coding region, which if you do the math is around 42 bases in length. And this coding region codes for a peptide, which is 14 amino acids long. And this is called leader peptide. We call it leader peptide because it is part of this leader region. Now in this leader peptide, there is a short subset of sequence that we will call region 1. After the stop codons, there is a region 2, then there is a region 3, and then finally a region 4. These squiggly lines here mean that there are bases which are not part of region 1, 2, 3, and 4. So finally, you have a stretch of 7 uracils. This poly-U sequence is at around +139 position. This poly-U follows up with another Shine-Dalgarno sequence, which is the important part because it comes right before the start codon of the tryptophan E gene. And this is at position +163. And the RNA continues into D, C, B, and A genes beyond this point. This poly-U sequence is the attenuator sequence that we highlighted previously. So to summarize this, the leader region contains a 14 amino acid coding leader peptide, the region 1, region 2, region 3, and region 4. And it also contains a short attenuator sequence. By the way, attenuator is just a fancy word for a termination signal. So that is the leader region, which happens to be around 140 bases in length. Now we're ready to discuss the functioning of tryptophan Operon itself. The major determinant of the Operon function is the RNA secondary structure, which influences the translation and transcription. And these secondary structures are formed by region 1, 2, 3, and 4. Let's get concrete and actually see the secondary structures formed by these regions. For this, we will focus only on the leader region 1, 2, 3, and 4, and the attenuator sequence. The regions 1, 2, 3, and 4 have complementarity among each other, which means they can form hairpin structures. In steady state, they can form a dynamic hairpin that looks something like this, where the attenuator is not part of the hairpin but sits at the end of the region 4. The region 1 and region 2 can pair with each other, and region 3 and region 4 can pair likewise with each other. But if you look at region 2, it pairs with both region 1 and 3. And likewise, region 3 pairs with both region 2 and region 4. So internal regions have two pairing partners. And this is what a steady-state dynamic structure looks like. This dynamic structure suggests that there are two possible equilibrium states, and we can draw them out. One state exists where region 1 and 2 interact. If region 2 interacts with region 1, then region 3 must interact with region 4. So the first state looks something like this, where you have two hairpins. In this state, the attenuator sequence is right next to the hairpin. The second state is when the internal regions 2 and 3 form a hairpin. And if this happens, the region 1 and 4 are left alone because they don't have any complementary sequence to form a hairpin. In this second state, the attenuator region is nowhere near the hairpin. And just remember that region 1 contains the UGG codons, which overlap with the leader peptide. Now we're ready to understand how tryptophan operon works in the presence or absence of its substrate. Let's start with a case when there is little or no tryptophan in the cell. Well, if the cell is low on supply, then it must make tryptophan. If there is no tryptophan, then by default, the repressor is inactive. And this means it cannot bind the operator sequences in the DNA. Which means at the repressor locus, the polymerase will bind the promoter, which will make the mRNA, and this will be translated into a protein. But at the end, you just keep making the repressors that don't do anything. If the repressor is inactive, the polymerase can also bind the promoter on the trpOperon locus and make the RNA. Alright, we have very low or no tryptophan in the cell. This implies that the tRNA that is charged with tryptophan is also very low in concentration in the cell. This piece of information is key to understanding the entire tryptophan operon concept. Let's get into the actual mechanism on how all of these fit into the bigger picture. Now imagine this, the RNA polymerase starts moving from the promoter of the operon and transcribes the leader region. And as the leader mRNA is made, ribosomes start translation from this mRNA, as soon as it finds the Shine-Dalgarno sequence on the 5' end of the mRNA. The ribosomes then move forward. To remind you, this scenario is possible because there is no physical compartments or organelles in bacteria, so translation can occur co-transcriptionally. So the ribosomes and RNA polymerase are moving in the same direction. But when the ribosome reaches the UGG codons in the leader peptide, the ribosome will pause at these UGG codons. This is because UGG codes for tryptophan, and since we are low on tryptophan, the corresponding tRNA is also low, so the ribosome cannot move forward. However, the RNA polymerase keeps moving ahead, and here lies the ultimate key to the tryptophan operon. You see, the UGG codon where the ribosome is paused overlaps with region 1 that we discussed earlier. This means that region 1 is not available to pair with region 2 because region 1 is blocked by the ribosome. And due to this block, region 1 and 2 hairpin cannot form. And as a result, region 2 and region 3 will come together and form the hairpin. This is exactly like this second state that we talked about. In this state, the attenuator is quite far away from the hairpin. This means that the attenuator or the termination signal cannot stop the RNA polymerase. So it continues into transcription. Now, here's a simple question to ask. What is the fate of the ribosome in this case? You can pause and try to come up with an answer. One possibility is that the ribosome falls off because it cannot move forward. Another possibility is that if the tryptophan concentration is low, the ribosome will slowly but surely reach the end of the leader peptide. It'll find the stop codon UGA and the translation ends there. The ribosome then dissociates at the end of the leader peptide. This small translation stint produces a small peptide, which is 14 amino acid leader peptide. Here's another question that pops up. If the ribosome falls off at the stop codon, how is it that trpEDCBA proteins are made? To answer this, you have to notice that the RNA polymerase moves past the attenuator signal. And downstream of the attenuator is another Shine-Dalgarno sequence onto which the ribosomes can bind and start translation of the downstream genes. So translation of each of these genes of these mRNAs will make the respective proteins. These proteins and enzymes then synthesize the tryptophan, This is exactly what we said in the beginning. If the tryptophan concentration is low, you need to make tryptophan. Alright, let's switch things around and take the case when the tryptophan concentration is high. If the tryptophan is high, then there is no need to make more tryptophan. It would just be a waste of energy. Specifically in the context of this operon, the expression of the trpEDCBA needs to be turned off. Let's start with a simple thing. The tryptophan amino acid has affinity towards the repressor. It binds this inactive dimer and makes it active. Since the repressor is a trans-acting protein, it can bind the operators at both these locations. The operators in question here are similar to the operators that we saw in the lac operon because they have a dyad symmetry, so they have inverted repeats that are not perfect. But the actual sequence of the DNA is different. The binding of the repressor is quite similar to the repressor binding we saw in the lac operon as well, except that you do not form loops in this particular case. Here's a funny thing about this repressor. The active repressor does bind at the operator of the operon, but its binding is not strong enough to stop the RNA polymerase, so it does not actually do much. On the other hand, when this repressor binds at its own locus, it is able to stop the RNA polymerase, and as a result, the repressor mRNA is downregulated, but the RNA polymerase on the trpOperon is unaffected, and it still makes the mRNA. So how do we stop the RNA polymerase from making the RNA on the trpOperon? To understand this, let's get back to this picture when the RNA polymerase is in the process of transcription, and it has just transcribed the leader peptide region. Again, just as we saw before, the ribosomes will bind at the 5' end of the RNA and start translating the RNA into a protein. This time, as it approaches UGG codons, the ribosome will not stop, because remember, we have a high tryptophan concentration in the cells. This means that the tryptophan tRNA concentration is also high, so the ribosome does not get paused, which means that the ribosome quickly gets to the stop codon, and then translation terminates and the ribosome dissociates away. This means that the 14 amino acid leader peptide is produced very quickly. But wait, the RNA polymerase is still moving, right? Well, not for long actually. Let's get to the crux of this. There are two possibilities in this situation. One possibility is that once the ribosome reaches the stop codon, it stays there for a while. And while it is at the stop codon, it is blocking region 2 as well. This means that region 3 and 4 are free to form secondary structure. So the region 2 is blocked either because the ribosome takes its sweet little time to dissociate away, or it reads through the stop codon. Both these conditions result in blocking of region 2, which gives region 3 and region 4 the possibility to form hairpin structures. The other possibility is that ribosome dissociates soon after terminating translation at the stop codon. In that case, region 1 and 2 are immediately freed up, which means region 1 and 2 can form a hairpin, and regions 3 and 4 can come together to form a hairpin. This is similar to the situation that we saw here. Keep in mind that the association of region 1 and 2, and likewise 3 and 4, occur because 3 and 4 hairpins form first, which leaves no choice for region 2 but to pair with the region 1. Also, this hairpin formation is timed in such a way that the moving RNA polymerase is very close or almost overlapping with the attenuator sequence in both of these situations. And it is the association of region 3 and 4 and the secondary structure formed by them which is the ultra important point. And I will stress this point again, that the timing of the hairpin formed by region 3 and 4 must match the position of the RNA polymerase being at the attenuator sequence. So you may ask, well, why is this timing and positioning of the RNA polymerase so important? That's because a strong hairpin next to a poly-U stretch, which is the attenuator sequence, means that the RNA polymerase will backtrack and slip off the DNA, like a train derailing from its tracks. We have discussed the details of this in the intrinsic termination of prokaryotes, also known as row-independent termination of transcription. You can check that video, link is down in the description if you want more details. Anyways, if this happens, the RNA polymerase therefore cannot move forward in transcription, and this releases the polymerase from the DNA. This means that the operon is turned off when the hairpin forms at the regions 3 and 4. This is exactly what we said was the logical expectation. When tryptophan is already high in concentration in the cell, you need to stop the Operon activation because you don't need to make tryptophan. Now let's get a little more technical with the terms here because it comes up in textbooks, research papers, and it may also appear on your exams if you're prepping for it. First is the idea of a repressor, which is a protein which directly decreases transcription. We saw this to be the case in the lac I, in the lac operon, where the protein is active by default. Then there is the idea of an aporepressor, which means that the protein requires additional factors before it can do its job. The job is, of course, to decrease transcriptional output. The additional factor here is called a corepressor. Now the example of this aporepressor is the trpRepressor, which is inactive by default. And it needs tryptophan to make itself active. So tryptophan is the corepressor. The combination of aporepressor and corepressor is called the holorepressor, which is the functional repressor. This concept is analogous to the holoenzyme concept. For instance, in bacteria, the RNA polymerase and sigma factors together form the functional holoenzyme. So aporepressor and corepressor by themselves are not functional, but when they come together, they make a functional product. Alright, so far we have talked about the regulation of tryptophan operon in E. coli bacteria. But it turns out that there is another type of mechanism to regulate trpOperon. This one is found in Bacillus subtilis. It uses slightly different components. The trpOperon in Bacillus subtilis also has an extra gene called trpF. The trpF encodes for indole 3 glycerol phosphate synthase, which is involved in an alternative synthesis of tryptophan. But that's not even the interesting bit. The tryptophan operon in this specific bacteria has no operator sequence. It also has no leader peptide in the leader region. The aporepressor trpR gene is also missing. Then how is this bacteria able to regulate tryptophan operon? The regulation in this bacteria depends on a protein called TRAP, which stands for tryptophan attenuation protein, also called tryptophan RNA binding attenuation protein. The TRAP is encoded by a gene which is part of the MTR operon. The MTR operon has MTRA and MTRB genes, and the MTRB produces the TRAP monomers. I will not go into the MTR operon here. All I want you to take away from this is that the TRAP protein production is independent of the tryptophan operon here as well, just like we saw in the case of E. coli. And this TRAP protein is an RNA binding protein. In this case, the MTRB RNA is translated into a protein, which is the monomer, for the TRAP protein. And 11 of these monomers come together to form a ring-shaped complex, which is the TRAP protein. But this TRAP protein is inactive. So technically speaking, this is the aporepressor in this case. And let's turn to the operon structure in this bacteria as well. Following the promoter, you have the leader region, but this leader is different because it does not contain any leader peptide. So no UGG codon dependency and ribosome pausing in this particular system. But there are 4 regions that have the potential to form hairpin structures, like we have already seen in the E. coli. The difference here is that regions 2 and 3 partially overlap. The organization is such that region 1 and 2 can form hairpin, and regions 3 and 4 form another hairpin. Regions 2 and 3 cannot, however, form any hairpin. This is in contrast to the structure that we have seen in E. coli. Anyways, following this leader, you have the E, D, C, F, B, and A genes that are involved in the tryptophan synthesis. One peculiar property of the leader region in this system is that it has a bunch of GAG and TAG repeats in the DNA, which means in the RNA it is GAG and UAG repeats. The RNA from this operon therefore looks something like this. A lot of GAG and UAG repeats in the beginning, and then an attenuator, and finally genes. Now let's see how the operon is actually regulated. In the first situation, say the cell is low on tryptophan, which means it needs to make more of this amino acid. So the operon needs to be turned on. In this situation, the TRAP is inactive. Well, that is the default state of the TRAP, because it is an aporepressor. So when the operon is transcribed, the RNA is made, the region 1 and 2 are transcribed before region 3 and 4. Hopefully that is not difficult to see. So if region 1 and 2 are transcribed before 3 and 4, then region 1 and 2 will already form the hairpin structure. This means that region 3, because it overlaps with region 2, cannot form a hairpin structure with region 4. This means that the Shine-Delgarno sequence, which is part of region 4, is free and accessible by the ribosome. So translation can start. And the attenuator is not sitting next to any hairpin. This means that the translation and transcription can proceed normally. The operon therefore is turned on. This is exactly what you would expect when the cell is low on tryptophan concentration. It will turn the operon on and make tryptophan. Now let's consider the case where you already have a lot of tryptophan in the cell. And when you have a lot of tryptophan in the cell, the amino acid binds the inactive trap protein and makes it active. And this becomes the hollow repressor. This is similar to the trp dimer of E. coli, where you need tryptophan to make it active. Here's the interesting bit. When the trap is active, the GAG and UAG repeats in the RNA are like the binding sites for the trap protein. Because remember, trap protein is an RNA binding protein. The RNA therefore wraps around this active trap hollow repressor, and specifically region 1 and region 2 are caught in this trap. But the end part of region 2, the part which overlaps with region 3, is not trapped. This means that region 3 can form a hairpin with region 4. And here lies the beauty in this system. The region 4 overlaps with the Shine-Delgarno sequence. So forming this hairpin means that the ribosome cannot access the Shine-Delgarno sequence. On top of this, the region 3 and 4 hairpin is next to the attenuator sequence, which means that the RNA polymerase transcription will intrinsically terminate as well. This system cuts both ways. It stops the transcription as well as the translation initiation. To circle back to the argument, when the tryptophan is high in concentration, the trap is active and the operon is turned off. And this completes our comprehensive discussion on tryptophan operon.